Hello everyone, welcome to the next video. In this video, we will be discussing the basic concepts regarding genes. Those who are new to the channel, let me tell you that we are going to have multiple videos on various topics of biochemistry, and I hope these videos, they're going to help you to understand the concepts in molecular biology, biochemistry. The first video that I made included some of the important concepts and molecular processes of the cell. And I briefly explained what we will be discussing in the near future. And this is the second video. What you can do, you can also make notes while I'm trying to explain few things. You can also take a screenshot of this particular area that is where I have written all these points, and I'm sure that these points will help you to understand the concept, and I've especially picked out the important points on that particular topic, and at the same time it will be a little bit elaborated, so it's not going to give you the very simple information. It will give you some sort of the advanced information that will help you in your project, that will help you in your homework, that will help you in your exams. So as you can see here, it's a simple format. I have written few of the things. I'll add up few things and also explain what is there on this slide, right? So without any delay, we should be starting with the topic. So as you can see here, what we have is basically the genes. That is what I have written, the name of the topic, genes. So what is the concept of gene? That we'll try to understand. Who are the people that were working on this particular concept, and what was their work, how they have identified the molecular definition of the gene, and these are the points we will cover. As you can see here, the first point that is written here, genes and codes. So genes basically is a segment of DNA molecule. If we talk about one gene, it's a segment of DNA molecule that encodes something. So traditional definition, if we consider, we can say it can encode a character, right? So I can write it down. I do have my other slide where I have some pointers that I'll take the help from, because it's very difficult for me to remember everything, right? So please bear with me. And as you can see here, okay, so first point as gene encodes something that will lead to a phenotypic character. So I can write phenotype, I guess, phenotype, right? And phenotype will come from where? Phenotype will basically be a character, I would say, and that will be coming from the cell, and there is molecular processes going on. New molecules are coming up. That is why you'll have new phenotype coming on. And then phenotype, as well as also nowadays we also consider phenotype that will indicate some sort of a protein molecule or RNA molecule, or an RNA molecule in this case. So that I think that includes the complete definition of the gene. Now moving on to the second point, which is written here, genes, what they do. So let me encircle this again. So genes basically, they control the character, a single character. Only one single character is being controlled by that particular gene. And in this case, probably we can take the example of the eye color, right? So I can write it down, eye color. And I can even draw eye so that you can basically remember what I'm saying. Oh, that's a really angry looking eye. Anyways, you understood what the point is, right? So sometimes you have different colored eye, dark brown, blue colored eyes, and so on. Green colored eye, this seems like a green color eye. So that's the one character that can come from the gene. Okay, what is the next thing? Moving on to the next one is, I have written two names, George Beadle and Edward Tautam. So you need to remember these two names. You can't just forget these two names. These are highly important, and we need to understand also what they have done when the work was concluded. So in this case, okay, the eye is without pupil. So let me design the pupil also, so it will look a little bit decent in that. No, it's still angry looking guy. So sorry about that. Okay, moving on to the third point. So the first point is the definition of the gene. Second point, again, explaining the gene in more detail. And the third point here, people who actually work to find out what is the role of the gene, or I would say the definition of the gene. So Beadle and Tautam, what they have done, they are responsible for the molecular, I'll just write definition, DIF, molecular definition of the gene. Okay, and how they concluded this, let me explain. And when it was done in 1940. So that's the year where they worked out the definition of the gene. Okay, what they have done, they took this particular fungus. So if anyone asks you that what was the organism, experimental organism for Beadle and Tautam, you will immediately answer Neurospora crassa. So this is one kind of a fungus. So here it has been written, it's a fungus, and what they have done, they expose these spores, okay, fungal spores. So that is what I'm designing in these small circles. I will also write it down here, spores. And they exposed these spores to X-rays and some other reagents, other agents, I would rather say that, other agents. And what are those other agents and significance of the X-rays? So what they're trying to do is basically trying to expose the fungal spores to some sort of DNA damaging molecules or DNA damaging radiations. So obviously X-ray, they will have an impact on the DNA. Other agents that they are using, they are also going to interfere with the DNA. And then because of that, what is happening is the outcome, right? So here I have written the outcome of that experiment was generation of the mutants. And mutants, when they were generated, it was found out that one of the key enzymes, so I need to write it down here, enzyme. One of the key enzyme is missing from those mutants. So when we compare the wild type, so if I say wild type versus mutant, what I mean by that is the spores or the organisms that are not being exposed to the X-rays and the mutants that are exposed to the X-rays. And they found out that in case of the wild type, you have all the enzymes needed, and in case of mutants, one or more enzymes they were missing. And sometimes, this is important, the entire metabolic pathway. So that is why I'm saying entire metabolic pathway was affected because of that absence of the key enzymes that are involved in those metabolic pathways. So they also identified that sometime what happens is some of the metabolic pathways, they also affected by the mutation. Okay, now I think I need to move a little bit down, so I'm going to scroll down here, and we're going to come back and again revise what we have learned. But okay, now this time I have to move. So the definition of the gene, they concluded, right? So this is the next point. Definition of the gene is basically a gene that encodes, but gene will be the segment of DNA molecule. So this is one part of the definition, segment of the DNA molecule, because the information will come from the DNA that will encode what? An enzyme. So this is one of the important hypotheses, and it is also known as one gene, one enzyme, right? Hypothesis. So this is one of the key hypotheses. Later it was found out that it is one gene, one polypeptide. And if you recall my first lecture, what is polypeptide? Is basically one molecule made up of lots of amino acids. Obviously, it is not going to be linear like this unless it is denatured, right? Otherwise, it's going to be secondary structure like this. Okay, so that will be the one gene, one polypeptide hypothesis. So this is how they worked out on this concept, and then when they saw the enzyme is not there, so they said if one gene is being affected, that may lead to the defect in the production of enzyme or one polypeptide in general. Because not every time genes they are going to produce enzymes, there must be some structural proteins, there must be some other types of proteins, or even an RNA molecule that can be produced. So this is important concept, right? Later on, they identified, or the researchers, they identified rather, that there is the RNA molecule also involved. So modern definition, now we are moving on to the modern definition of gene here, right? Okay, now moving on to the modern definition of the gene. Okay, now you can see what I have done is just there are some fill in the blanks that I'll try to cover, and in the meantime I'll also explain those things. Modern definition is basically gene, right? In the older definition, we know that it's a segment of DNA. So segment of DNA needs to be there, right? Which encodes, it is going to encode the polypeptide. So yes, that's the case we have, right? So we do have the polypeptide word here. So I can write polypeptide, and I've already explained what polypeptide is. It's nothing but the chain of the amino acids. Now polypeptide can be there. What extra thing that we found out later on is the RNA molecule. So I can also write this, that it can also contribute for the formation of RNA, and RNA can be of structural importance, it can be of functional importance. So these are two important terms that we need to remember in this case. Moving on to the next concept, which is regulatory sequence. So I just thought that I should put this particular point here, that regulatory sequences are also there. So that means it's not always important that you're going to have DNA molecule that is going to encode protein or the RNA molecule every time, but there should be those regulatory sequences that can control the expression of the gene. So what they provide is basically the signal that will lead to the controlling of expression of the genes. So they are the regions of the DNA where nothing is going to be coded. They are the region where proteins can bind, and based on that, they are the region they will control the expression of the other genes, right? So they can be involved in the initiation. So I will just write it down. Initiation of the expression, or they can be involved in termination, right? Termination of the expression. Expression here I'm writing is basically nothing but the gene expression. Correct? So this is important, that we do have regulatory sequences, and that is not being included in the definition of the gene. So that is why I intentionally edit that. So I'm moving down. So this is covered. Next, multiple genes. Another important concept is sometimes one stretch of a DNA sequence can also give you multiple genes. So I can write many genes. That's one point. Many genes are coming from a single sequence of the DNA molecule. So this we need to remember that one segment, rather it should be written one segment of DNA molecule. And how this processing happens, we will study that process in detail in the later topics, but not here. But you need to understand that many genes, they can come from a single segment of the DNA molecule, and for that one basically what will happen, there will be combination of different segments, and then you will get multiple variants of that particular gene. moving on to the next one is some calculations. So I think I have seen some of the numerical problems coming out from this particular concept. Here we need to understand that, okay, one amino acid basically will be coded by 3 base pairs, right, of the DNA molecule. So this is important. For example, AUG, you will have one amino acid. It can also act as initiation codon, right? So that is important point. Based on that, you can get the question that how many, for example, 350 amino acids, how many bases of DNA required for this one? That probably 105, a 0 base pair of the DNA is required for this particular amount of amino acid. So it's a polypeptide molecule that can be produced by these base pairs in a DNA segment. Moving on to the next point, which is coding and the non-coding regions. Coding and the non-coding regions. Okay, so this is a very, very important concept, and I thought we should be discussing this, and we will be talking about these things. So coding region is also known as exons, and non-coding is also called as introns. Okay, so these terms, please you need to remember these two terms. What are exons and introns? Exons are the segment of the DNA that are going to code a certain polypeptide molecule. Introns are the regions in between the exons. So if I draw a segment of DNA like this, you will have small regions somewhere which we can call exons, and some regions which we can intron, and they will be interspaced between different exon regions. And the role of intron regions is basically not going to code any specific amino acid or a polypeptide molecule. They need to be spliced, and still research is going on understanding what are the roles of these intron regions, and we have identified a lot of important roles of these intron regions as well as. But right now most of the research is focused on the exons. Now what is exon? Exon is nothing but the DNA sequence. It's also a DNA sequence. Intron is also a DNA sequence. Exon is also a DNA sequence, so that is common. Next is that will code. So code word is important. That will code for the protein or the polypeptide or the RNA molecule. It is not going to code anything. So these are the two important features or differences between these two. Now another important point is introns, they are there also in case of bacteria and in case of archaea. In very, very few examples are there, but in case of eukaryotic organisms, you will see introns quite often. Okay, now moving on to the next point, I'll throw out some of the important numbers here, and I think they will be important for you. So please remember those numbers, and you will be benefited because direct questions in the exams can come from these topics. Here, the number is coming from E. coli. So this number is coming from E. coli. E. coli is one of the well-studied bacteria. It is being used in genetic engineering. We have studied different strains of E. coli in very much detail. We have studied those organisms because E. coli is one of the beneficial organism, so it's not causing any disease. It's easy to handle. A lot of bioethical complications are not associated with this organism, so it's easy to work with. We have studied E. coli, we have sequenced E. coli, and we found out how many, look at that, how many genes are there. Probably I used different color to show those numbers. So you have 4, 6, 3, 9, 6, 7, 5 base pair of the E. coli DNA. So E. coli DNA have so many base pairs. And how many genes are there? You have a total 4,300 genes plus 153 genes that are going to code for some sort of catalytic or functional RNA. So I can write it down. I have a very small space here, and these are going to code for proteins. Here, functional or structural proteins, and then you have 153 genes coding for the RNA molecules, structural or the catalytic RNA molecules. Now, this is the circular DNA molecule. It's not obviously entirely circular, but this is the number. You have 4, 6, 3, 9, 6, 7, 5 base pairs of the DNA, in E. coli. So this we have to remember, right? So this is the structural or functional RNA molecule, and genes I have already mentioned, 153, in this case 4300. Now in case of human genome, right, we need to understand this because now we are talking about us. How many genes? I have done my research. I found out approximately 25,000 genes, and 20 to 25,000 genes are there in case of human. And how many base pair? 3.1 billion. Okay, we're talking about humans. We have 3.1 billion nucleotides in our genome inside a single cell. So we are talking about one cell. If you are not familiar with the structure of cell, I recommend you to watch my videos. Inside one single cell, how many base pair of DNA molecule is there? 3.1 billion base pairs. Look at that. Look at that. How many molecules are there, right? So, and that is why you're producing a diverse pool of genes, 25,000 genes, and they are working in a synchronized way, and that is why our body is functional. And then if there is any defect, we are definitely going to have diseases. Okay, so this is the significance of the gene expression. We have studied what are the different definitions, how we came to the conclusion, the modern definition of the gene, and also some of the numbers I talked about, right? So if I go quickly move to the start of this lecture and quickly go down, okay, that scary looking eye is still there, right? I hope you remember what was the example for that. So we started with genes, and then we understood what gene encodes, the definition, modern definition of the gene. I gave you the example of phenotypic character, and that is in case of the eye color. Moving down, we talked about the work that Beadle and Tautam did in 1940, and then we identified that here, we identified what is the ancient or the primitive definition of the gene, and then we moved on to the modern definition of the gene in a sequenced way. Moving further, regulatory sequence we identified, and then multiple genes, the concept of multiple genes, and then we moving on to the calculations. Sometimes you get these questions. It's a very, very simple. We'll talk about more complex questions in our later videos. Then we talked about exons, introns, and also about the E. coli as well as the human genome, genes, as well as the total number of base pair. Right? So I think that includes most of the important concept regarding the gene, modern definition of the gene, and nowadays what we consider is a gene and why it is important, how the expression is being controlled, and what are exons and introns. Okay, so I'll just conclude this video here. Please watch the videos in that sequence, because we'll have every chapter is being discussed in detail, and if you break the sequence, then what will happen is you are not going to understand the topic that is being taught. So watch the video like episode 1, 2, 3 in that manner, and you can also make notes. You can also take snapshots. So this video is also going to be helpful in providing you the snapshots, right? And you can also see the videos in that order and trying to understand the entire chapter. All right, I'll meet you in the next video where I'll try to pick similar topic and extend it, and I'm sure there are so many topics still in this particular chapter. This is the first one that we are covering. We'll move on from replications. Replication will move over transcription, translation, regulation, and all those important concepts we'll be covering in our video series. So thank you so much for watching the videos. Thank you so much for your support, and I request you again, if you haven't subscribed to the channel, then please do subscribe to the channel. And if you have any questions, please feel free to put your questions in the comment section below. I'll try to answer them. All right. Thank you and take care.